Okay, so differential gene expression. I'm sure you're all familiar with the concept already, but there are a lot of things to consider when you do differential gene expression in single cell compared to bulk. So today I'm planning on first talking about what differential gene expression is, just so we're all on the same page, and then how to perform the analysis, both in choosing your groups of interest and what to consider there, which functions you can use to run it, what it means when you have a simple design versus a complex design of your study, and then the differences between running 1 versus 1 and 1 versus all comparisons, and then some other considerations that I don't cover in the previous parts. So first of all, what is differential gene expression? You're all aware that what we're trying to find in this is genes that are more highly expressed in one group compared to another. So for example, we might have something like this, where one, in this case cluster, clearly has higher expression of one gene compared to another cluster, or lower compared to another cluster. And then sometimes you'll have or most of the time you'll have genes where it's either clear that there's no difference or unclear if there is a difference and how important that is. And so to perform differential gene expression analysis, we need count data, we need to perform some sort of statistical test, and then we find out whether the differences are significant, i.e. Whether the differences are greater than what would be expected if we just random sampled the population. So a reminder about the statistics. When we do a statistical test, we have some sort of null hypothesis, and this varies between methods. But in general, we try to see whether group A is equal to group B, and that can be in mean or median or distribution. And the p-value says that if the null hypothesis is true, we can expect the measured result or something more extreme in less than, so in this case, 5% when p is under 0.05. Where we have sampled group A and B with the sample size that we have. So for example, if the original populations have a distribution that looks like this, if we sample them, we might see something like this. And then we need to figure out, just based on this information, without knowing this, how likely it is that this actually came from two populations that are the same. And the p-value gives us the statistical significance, and I highlight this because, so this means that the result is likely to be from a true difference rather than random chance, but it does not say anything about the biological significance, so that you still have to figure out for yourself. And the main output of differential gene expression is the same as with bulk, that you generally have some sort of effect size parameter, in this case, the fold change between your groups. So for fold change, if it's, or log 2 fold change, if it's positive, that means it's higher in one group, and negative, it's lower in that group. And then you have the p-value, and because we're running so many genes, like checking so many genes at the same time, we need to adjust for it. Or we need to adjust for the number of tests. And so you will get a p-value as well, but the adjusted one is the one that is important to look at. But in single cell RNA-seq, like Åsa mentioned, there are going to be a lot of zero values. And very often, rather than having these nice clouds that we saw earlier, we'll have something more like this, where in cluster 1, there are a lot of genes that express the gene, or a lot of cells that express the gene. And then in cluster 2, we have fewer cells that express the gene. And this can be visualized a bit better with the violin plot, where we see that there's clearly a larger proportion in cluster 1 that express this gene compared to the other cluster. And this is something that we need to remember in single cell RNA-seq, that whenever you see a log 2 or like a difference in mean or something like that, it can be based on different things. So for that reason, you also get a table with the percentage of each group that express the gene. Typically, it's just above 0, and this, they say percentage, but it's actually proportion. So in this case, 70% of the cells in one group express this, whereas only 6% in the other group. So in that way, you can see whether this is something that is very homogeneous for your group, or if it's more of a mixed population. But in addition to differential expression and proportion, there are also other types of differences in distribution. And since we have the single cell data, it would be good if we can also see those types of differences. For example, you might have that they have the same mean, but they have different variability, which can be interesting biologically as well. Or they just have completely different distributions, but with the same mean. But most methods that we have today focus on these type of differences, mostly looking at the difference in means or medians. And so in addition to not all differences showing the difference in means, you also need to consider that a difference in means can mean a lot of different things. So now that we know what we're looking for, we need to figure out how to perform the analysis. So the first thing to do is to define your groups of interest. And this can be a priori defined groups. So for example, you have cells from different samples, you have different experimental groups, like different treatments. You might have sorted cells, which is also same different samples from sequencing, but they can be different, like from the same original sample. And these differences tend to be more subtle compared to the other option. So you might have a UMAP that looks something like this, where you have your clusters and your groups are mixed in there. The other option, which we will do as the first step in the lab, is to do your differential expression between your groups that you've defined using the data. So when you do this, when you do a differential expression analysis between clusters, for example, and this can either be based on the RNA that you will also be performing the differential expression on, or you might have your identity from other data, like if you have multi-omics, you might define your groups based on the protein and then look at the RNA. In the first case especially, you need to be a little careful when interpreting it. So when you perform differential expression on clusters that you defined on the same data that you perform DA with, you can call that double dipping the data, then that will inflate the DA analysis. You'll almost definitely get some results because you've already seen the differences when you clustered it. You've seen that there are differences. Yes, question.
Hello, Jennifer. I have a question regarding like this UMAP on the right. There are some cells from which fall in different cluster sets. So when you do differential gene expression analysis, do you remove these cells before you run the differential analysis?
I typically wouldn't because, as also as mentioned, the algorithms for clustering in UMAP aren't the exact same, so this will happen to some extent, and it's like both of them tell some story, the UMAP and the clustering. If you find that it's a large proportion, then you might want to go back and redo your clustering or UMAP to see why they're doing this. Maybe you need to filter again. But in general, if they've been clustered in one cluster and then the UMAP looks like another cluster, they tend to be like they have aspects of both. And if that's enough to like shift your differential expression, then you should probably go back and check why that is.
Thank you.
Could you please explain the concept of double dipping? Because I don't understand what you mean by the same data.
Yeah, so if you use do the clustering on the RNA, and then you perform the differential expression on the RNA, those you need to remember that what you're comparing, like we tend to see it as, okay, well, we see that these are T cells in this cluster and these are B cells in this cluster. And then when we do differential expression, we say, oh, T cells have this gene, whereas B cells have this gene. But that might not be the case. It might be that they've clustered based on something else, based on this gene, and that it doesn't really have the same biological relevance. It also has to do with like like what population are you actually testing when you do this? If it's reasonable to, or like how to classify that population truly. Okay, great. So yeah, that was how to define your groups. And we'll get into a bit more when the groups become more complicated. But first, we'll talk about how to run this type of test. And so for all three of our toolkits, we have functions that make it easier to run the test. These aren't functions that have the statistical test like as part of the function. It's more that it can apply the function onto many tests or many genes. So you only have to run one command. So in the example of SURAT, we have two because this has to do with whether you're comparing one sample to another or one sample to many others at the same time. So in the examples of, in the example of final markers, you have a lot of different parameters that you can adjust. These are a little less, like they will affect your results a bit less than all of the parameters that you can choose in the integration and UMAP than that. But the important ones to keep in mind are this assay and the slot. So the assay is typically going to be, I mean, it will be applied to your default assay unless you specify, and that will typically be your RNA. But you need to make sure that it's not your integrated data if that is its own assay. So it'll usually be the RNA assay, but you can also run it on, if you have multimodal data, you can change which one you're applying it to. And then the slot will be, if it's data, then it'll be the normalized counts. If it's count, then it'll be the raw counts. And this depends on which test you're planning on performing. And then you have some parameters where it's mostly just to speed up your process. So you can change, you can filter the genes from the start, saying that I'm only interested in genes where there's a log fold change of at least 0.1, where it's expressed in at least 1% of cells in either group, those kinds of things. And that will just reduce the number of tests you're performing, so that instead of 10,000 genes, you've excluded 9,000 of those. And yeah, so it would be computationally faster. And then possibly the most important one is the test use parameter. Yes, question?
Hi, Jennifer. Yes, thank you. So you said that in the assay, this will be performed in the RNA assay, either in the normalized or in the scale data, but not in the integrated data. So integration is only for visualization then?
It's for clustering. Like for me, the integration is mostly for the clustering.
And then it doesn't really matter how we see it. We perform differential expression in the non-integrated data. So basically,
Yes.
we good for differential gene expression. We don't need to do integration at all.
If you're just gonna like if you have two samples that you're going to compare without clustering or anything before that, then no, you don't need to do integration. Then you can just go from the normalized data.
Okay, thank you.
Yes.
Can I ask you why that is? Why can you not run it on the integrated data? How would your data change if you would run differential expression on?
So
integrated?
largely it's that you're removing differences when you integrate. And so if you want to compare between, this is especially important if you're planning on comparing between your samples. If you integrated them, you've removed a lot of the differences. And so, but this is, what I was going to add before actually was that you can also then adapt this to correct for batches. And I'll show you how to do that in a later step. So if you have problems with like that you needed to integrate in order to remove batch effects, then you can add that as a part of your differential expression analysis.
Can I also just add a comment that most of these methods we use for integrating are not producing corrected count or data matrices. So there is nothing to test for differential expression. There's not a corrected gene expression from the integration. And then, of course, we have to go back to the RNA. We use the clusters we got from integration, but we use the raw RNA.
Thank you.
The next step is to choose your test. In Seurat, it's the Wilcox and rank sum test that is default. In the other two toolkits that we have, it's t-test that is default. Both of these two tests are very straightforward. You just run it on two different groups, and they're also very fast to run. But then there are some other options. So if you look at the help pages for the find markers functions, you can see which test you can use. This is Seurat's explanation of them. And so I said Wilcox and rank sum and T are the straightforward ones. But then there are some where they try to optimize the test according to the distribution. So the Wilcoxon test doesn't assume any type of distribution, whereas the t-test assumes normal distribution, which isn't actually the case in single cell, but it tends to work quite well anyways. But then there have been attempts to optimize the tests by using other distributions, and also including the DSEG2, so applying the same test that you would to bulk samples to single cell. It is a bit hard to know which distribution to use because the genes tend to have different distributions, and this will be based on a number of different factors. It can be noise, but it can also be depending on how differences in libraries between cells and also how deep you sequenced, it can be a low quantity, or it can be dropouts or amplification bias, and also the bursting, which causes, like also mentioned before, of the on and off, even in a cell that is producing the protein. And then also depending on how mixed your cell types are, you'll also have a different distribution because different cell types will have different distributions. And when you combine them all, it'll be a different distribution. And then there's another popular test that is what's called a hurdle model. And so the point of a hurdle model is to step away from this assumption that what matters is just the difference in means or medians, and also consider whether the gene is on or off. And so looking at the distribution of how many modes do you have. And the MAST is an example of that. So it's called a two-part GLM, a hurdle model, and it uses both the continuous gene expression and the discrete gene expression. And also, so it's able to handle dropouts, and it also supports complex modeling, which leads us into the next slide, where we have complex designs. So this is when you want to move away from just comparing one group of cells to another group of cells, but instead you have groups of samples. It's very common nowadays when we are able to sample from different patients and you want to compare that to a group of controls. You might have batch effects that you want to correct for, or other covariates that you want to correct for in order to find the biological signal of interest. And to give you an example of why this is important to consider, if we have an example of 3 patients and 3 controls where we have a lot of cells for each one, then the question is, when we perform our test, what is our N? Is it all of the different cells that we have, so possibly thousands, or is it the number of individuals like we're used to with this type of study? And if we just consider every cell as one replicate, then in this situation we would get a very low p-value for this gene, because you can clearly see that the distributions are different. But if we look at each individual patient and control that these cells came from, when we just look at this, we can very clearly see that this is just one patient that is driving this difference. And from performing the research, we know that if we just have one patient that is different, that doesn't say anything about the general patient population. And this is not just incorrect in terms of later interpretation, but the statistical test is invalid because the Wilcoxon rank sum, as well as other tests, they assume independence, meaning that they assume that all of these cells are independent samplings from the same population. But that's not the case. We've like some of these cells are expected to be more similar to each other than others. So, and also as an example, if you have covariates, for example, if you want to compare two clusters, and in this case, we have one cluster here and one cluster here, and we have two samples that these clusters came from, and we have the IL-10 knockout and the wild type, and you see that the composition of the cells in terms of clusters, they're very different between the samples. So if we look at the expression of the IL-10 gene, which is knocked out in the IL-10 knockout samples, in those samples it's 0, and then in the other samples it's expressed. And then if we just compare these two populations without considering the differences in compositions from this known covariate, then we'll get a significant result, even though when we look at each sample individually, we see that there's no difference between the two clusters. So those are examples of why it's important to keep in mind. Now, as to what we actually do about this, yes, question?
Yes, I'm sorry, but from my simple understanding, so the covariates should be independent from the statistical test, right?
What do you mean independent? I mean, they should be included in the statistical test.
Yes, but I mean, say they're not affected by the experimental condition itself. So I mean, say here, for example, when we have the knockout example here, so the knockout is the one that drives the differences in the samples, right? So in that case, it shouldn't be considered as a covariate.
Sorry, covariate is maybe not the right term, but it's like it's an effect that is not the biological effect that we're looking for. Because when we just want to generally identify what the differences between these clusters are, in this case, if we include both samples, then we'll see one difference. Whereas if we do the differential expression independently between the samples, like for one sample, and then for the other sample, we'll see other differences. So we can just, since what we're probably looking for is more what is different between the clusters, regardless of which sample they came from, then you need to include that information in some way.
Yes, exactly. That's what I mean.
But
It should be
this
independent from, yeah.
But this does depend on the question that you have. If you want to, if your question involves the sample in some way where, or maybe not in this case with the samples, but you might have some variable that does affect this, but that you also want to consider as part of the biology of it.
Yes.
But you need to consider a lot of factors in that case.
Thank you.
All right, so on to how to adjust for this. So basically, you can group the tests in a few different ways, but here we have 4 main groups of tests. So the naive model, or the simple design, is what you can just very easily run with the find markers functions. And in this case, we just assume that all of these cells are independent from each other, and all of these cells are independent from each other, and do they come from different populations or not? If you have some type of batch effect or sample effect, where you have groups that where both batches, for example, are represented, then you can use a generalized linear model, GLM, with latent variables. And so that will add this covariate to the design, and then it'll adjust for that fact. And so sometimes this will cause the differences to disappear. Sometimes it'll cause the differences to come out even clearer. So for example, here, even though the differences in means are the same between the batches, the variance is reduced when you look at each batch individually. So that will likely increase the statistical significance. The other option, or one of the other options, is a mixed model. So MAST has an option to include what's called a random effect. That's an example of a mixed model, where you include information about which original sample the cells came from. And so this will consider each cell one observation, but it will also retain this information that they are not independent between each other. And this you need to use, or you can't use the latent variables if you don't have shared groups or variables between your groups. If all of the samples in if each patient that you want to include, they're only present in the patient groups, so then you can't use the latent variables, but you can use this mixed model. And another option is to reduce your single cell information into essentially as if you did a bulk RNA-seq analysis on them. So you sum up all of your counts for all of your genes, and then you do a normalization for that to see, to generate a matrix that is similar to a bulk matrix. And of course, you lose the information about the single cells in this case, but you have a very, if you have a clear signal here, it's very reliable in general.
I have two questions, Jennifer.
Yes.
So in the generalized linear model, latent variables, you say that here, if we label batch 1 and batch 2, it'll increase our p-value.
No, sorry, increase the significance. So
okay, that I understand that. And then for pseudobulk, can we do this on specific cell types only? So basically, and I guess that's like the preferred way to do it. Like you lose cell resolution, but you still have the cell type resolution.
Exactly.
Right? Okay.
Yeah, so it's essentially that you can, instead of sorting beforehand and running bulk RNA expression, RNA-seq on all of the samples, you can start from the cells and then group them as if you were, sorting them. There will be, of course, it'll be more artificial because you've grouped them according to the RNA, which is a little less reliable than FACS markers and things like that. Yeah. But you can definitely do it on individual clusters. Yeah. And then you can also, when you have your differential expression, you can go back into the single cell resolution and check what the distribution looks like as well. Once you have your differential significant
genes. Basically, you could use this to kind of have a more certain repertoire of genes that are differentially expressed, and then go back to single cell level and see what is happening. Oh,
exactly.
Cool. Very cool. Thanks.
And I've made a little table, just very broadly summarizing. So don't take this as the gospel, but the four main approaches that I discussed, they have advantages and disadvantages. So in addition to what I already mentioned about whether they can include covariates and multi-level design, so this meaning that you have groups of samples. They're also different in the speed. So the naive models and the pseudobulk are very fast, whereas, or usually very fast, whereas the linear models and the mixed models tend to take longer time, especially the mixed models. And then they also differ in which type of false, which type of error they're more prone to. So this is especially if you're trying to use these models for a complex multi-level design test, then they will be very sensitive, but very unspecific. So you'll have a lot of false positives with those methods. Whereas the pseudobulk is criticized for being underpowered. So typically the genes that you get will be true, but then you will also have a lot of false negatives typically. And that's just because you're reducing your information, you will lose power as well. And then the mixed models, they're a bit in between. So which model you choose to use will depend on which error you're more okay with. And then also, for example, you might want to start with the pseudobulk to see if there's any differences there because it's faster, and then maybe do it with the mixed models as the next step. And I have some references here in the presentation if you want to look more into the different types. Then for how to do it when you have multiple groups. So this is not part of the complex design. This is just in terms of in which order you're testing things. So if you have, for example, 4 clusters, you can choose to either compare this cluster compared to each of the other 3 individually, or you can do this cluster compared to all other ones at the same time. Like you group these into one sample and then compare the 2. Some things to consider if you want to do that. The main advantage is, of course, it's fast and you have fewer lists to look through afterwards. But if you have an uneven distribution of clusters, or like an uneven distribution of cells in your clusters, then the larger clusters will be overrepresented unless you downsample first to have the same amount of cells from each cluster. So what this would mean is if you have cluster 3 compared to all 3 of these, unless you subsample, then a lot of the differences that you see will be the differences between cluster 1 and 3 and not so much between the other ones. And you'll see how to do that in the exercise as well. As for the second point, so highly similar clusters, for example, here, cluster 4 and cluster 1 might be more similar to each other than the other 2. Maybe they're 2 subtypes of a broader cell type, and whereas these 2 are their own cell types. And when you do the 1 versus all, then you'll often see that cluster 1 and cluster 4 have a lot of overlap in which genes are differentially expressed compared to the other clusters. And then you might want to look more at the 1 versus 1, comparing cluster 4 and cluster 1 specifically. Finally, some other things to think about. I want you all to be aware of the p-values being easily very small in single-cell data. This is partially because people use independent assuming tests when they shouldn't. But it's also just because we have a lot of cells and we're typically not used to working with with such a large N in biology. So remember that your P will depend on your N, so your sample size, as well as the variance and the intergroup difference. Here I just have an example of samples from different populations. So where this one and this one, they're from the same population, but with different N's. And this one is from another population with a larger difference in means. And I think that a lot of us, when we see a p-value like this, a very small one, we will tend to assume that there's a big difference in means. But if we have a lot of cells, then that difference in mean might be a lot smaller than we're expecting. And it's not just about means either, it's about standard deviation. So it's always good to check what your p-values actually are saying and not just look at the p-values. And to ask yourself whether all the statistically significant differences that you see, whether they're actually interesting to you, or if you need to do some selection of genes. Another thing that is easy to get a bit lost in, I think, in single cell is if you have 2 samples and they have different distribution of cell types. For example, here, this cluster is all from sample 2. And so if we compare sample 1 and sample 2, we'll see that some of the differentially expressed genes, although here it's not significant, but you get the idea. they might be specific to a cell type. So here, this is the neutrophil cluster, and so then the sample will have higher expression of the neutrophil genes. And this is true. This is not like a false result, but we always need to consider how we interpret it. Are we interpreting as a difference in composition, or are we interpreting as a difference in gene expression? And you tend to go a bit back and forth between what is more relevant to show. And then once you decide, often you'll decide, okay, well, then I'll just look at this cluster and see the differences between the samples in this cluster. But then you might also have subtypes, and is it then more important to look at the composition of subtypes or the gene expression? So just keep this in mind, that you're always looking to some extent at heterogeneous populations. And then probably the most important part is to always assess your results. It's very hard to say beforehand which method is going to be best. They're hard to evaluate in a general way. There have been a lot of benchmarking tests using data sets where we expect a certain difference, or simulated data where we even more expect a certain difference. But these might not be the correct data sets to use to compare for your specific data set. One way to assess the results is, like has been mentioned before, if you try multiple methods, then the intersect between them might be more reliable. Also, if you look at what is different, can you see why are they different, and does that make sense with the test? And then especially always checking your data, going back to, for each gene, going back to your distributions and checking, does this make sense? So you can get a lot of different distributions, and sometimes it'll be differences in proportion, sometimes it'll be differences in global population, sometimes it's just a few cells that are increasing the mean so much that the means are different. So violin plots are a very good way to see the full distribution. Another way that I think is useful, especially if you have more clusters, is to use the dot plot, where so this is a function in Seurat where it very quickly generates a plot where you have the color based on the expression, usually scaled, and then the size tells you the percentage. So this will also give you an idea of whether these cells that you find, are they global markers of your populations? Are they markers of a specific subpopulation? Are they expressed in all your clusters, but just a little more in one? So yeah, always go back to that. And then I have a long list of kind of summarizing most of what I've said, but with a few extra things. So when you start your study, and also when you're clustering and then performing differential gene expression on like a smaller group of cells, you need to consider how many cells and or samples that you need to trust your differential gene expression. And this will depend on a lot of things. So, for example, how different do you expect your cells or samples to be? If you're looking for very large differences, you probably don't need a lot of cells. And especially if, for the next point, if the expression is high in one sample and low in the other, or just high in general, then you can be more sure that when there is a zero, that it's, it's possibly bursting, but if there's a group that has more zeros, it can definitely be that there just is an expression. And this will also depend on how deeply you're sequencing. If you're not sequencing deep enough, then if you find a few reads in one sample and none in the other, it's hard to say whether that is actually relevant, or if it's just the random sampling. And then it will also depend on your library quality, especially because you need to filter out cells, but also just how well you can trust the results and how variable they are and things like that, and how well you can trust your normalization. When you consider which tests you want to use, start with thinking about which populations are you comparing. Are you comparing just populations of independent cells, or are you comparing patient populations? Are you comparing populations that you've defined before or after with the data? And then always check whether your cells are, or like think about whether your cells are independent and if you can use a test that assumes independence. And then also if there are any batch effects that you need to correct for. For which data to use, whether it's raw or normalized, that depends on the test that you choose to use. So check the documentation for that test. Always remember that differential expression results are relative to other cells. This is especially important when you do differential gene expression to see, to annotate cell types. If you're looking for a certain gene marker that is a marker for a cell type of interest, if all of your cells express that gene, like you won't see that gene in the differential expression, but perhaps that gene marker has been defined in comparison to other cells, so you shouldn't expect it in the differential expression here. Never just rely on the p-values. Always look at what the actual effect is, and then always assess your results. So visualize full distributions, especially if you want to claim that specific genes are differentially expressed. Like if you're using a specific result for further study, then always visualize it and check for potential confounders. So this is again going back and forth between your QC, your batch effects. Whenever you find a result, check whether it correlates with any of these other factors that could involve that could affect the result. To some extent, you might be able to correct for batch effects with latent variables. And then also, like also mentioned previously in the QC lecture, sometimes if you remove ambient RNA, you can remove some of the effects from like differences in populations. So in conclusion, remember that single cell data is more complex than just differences in mean expression. So going back to looking at distributions, the different tests rely on different assumptions. So consider those when you choose your tests. And also when you interpret the tests that other people have used. Always consider the populations that you're trying to compare. And this goes back to the different tests and their assumptions, but also just when you're drawing any type of biological conclusion, consider what you've actually been testing. So like mentioned before, if you're testing clusters, be careful with how much you're extrapolating that to biological cell types or something like that. And always important to assess and validate results. So the validation, since single cell is, or like bioinformatics is generally predictions and can lead you to the results or the genes that are most likely to be having an effect, but it's not until you can validate it biologically that you can really trust that it is important. In the presentation in the schedule, you'll also find this with the references if you want to look at anything further. And then I want to acknowledge that this presentation was built upon previous presentations by Olga Detlevson, Åsa, Vincent Van Hoff, and Roy Francis. Do you have any more questions? Yes.
Yes. So if we have a condition where we have actually multiple treatments done at one time, and then in one particular cell, and then cell type, and then let's say a knockout of this cell type with the same set of treatments that was done at a later time point. So in that case, we have a mix of the patch effect here because the preparation is not done at the same time. And then we have the complex designs. In each experiment, we have like 5 or 6 different treatments. So in that particular case, how would be the ideal test to do?
Yeah, it's a good question, and it's something that there's no like easy answer, I would say. If you have like it is always like if it is possible to change the experimental setup to sample them at the same time, that will help. Otherwise, like also dividing them up so that there's not like one batch effect in your experimental groups. But if this is the study that you like, the design that you have to work with, it is important to consider that that might be an effect that you see. If the batch is the same as your experimental group, it will be hard to say for sure that that it is biological and not a batch. But then you can also, when you interpret the data, you can see, does this make sense? And then you can be a little more, like then you can interpret a bit more. And again, as I said, it's always predictions. So it's like even if things are statistically significant or not, it's still you need to consider, does it make sense, and like is this something I can go forward with? But yeah, it's hard to correct for it completely.
So because sometimes in such particular settings, it's really hard to design an experiment where everything can be done at the same time. So it has to be
Exactly.
done in batches. So do you think first it should be corrected for the batch effect first? Then
It's not really like if the batch effects are completely overlapping, then you can't really do, like if the batch effects are, or if the batches are completely overlapping with your experimental groups, there's not really a way to correct for the batch without removing your differences between your experimental groups. If you have, yeah, I'm trying to think if like one way to do it is if you have, if you can split up even more and have more batches. It sounds counterintuitive, but then you can at least get an idea of how strong might the batch effects be, and are like if at least your experimental groups are comparable between your batches, then perhaps like between batch 2 and 4, then perhaps you can expect the difference between 3 and 4 to be smaller as well. Åsa, do you have any input?
Yeah, I mean, it's not super straightforward, and I'm not really clear on exactly what kind of design you're talking about now. But you should, and if you even have replicates of your different conditions or not across your batches, because if you do have replicates, so you have each condition is at least in 2 batches, sort of in your combinations, then you can use these sort of complex designs and include both batch and treatment information in your model. But if you're trying then to test between your different treatments, for instance, and that's like completely confounded with your batches, then, I mean, of course, you can use the mixed model, these mixed model like random effects, but you can never be 100% sure what is a batch effect and what is
biological effect.
biological. Then you need verification experiments to validate what you find.
Yes.
Like I say, like generally, it's very rare that you have a perfect study design. So the most important thing is to consider all of the things that could be influencing your results, and always remember those when you're interpreting, so that you can consider how likely it is to be due to, the study design that you have, and considering those restrictions, what is your best way forward?
Okay. Yeah, thank you.
Any other questions? Yes.
Yes, thank you. So you said that we might consider removing ambient RNA.
Yes.
So, and then, I mean, we talk about this in the first lectures, I think, first days.
Yeah, exactly.
Remember, we did it actually, but I know that SCT in T-RAT can do it, like at least estimate it. But do you recommend any specific tool for removing?
Åsa knows more about this. I
The
don't work with it much.
SCT has nothing to do with ambient RNA.
Okay,
Start with, just to make sure that we're on the same page here. So I mentioned in the lecture a few tools for removing ambient RNA. I would probably recommend Cellbender. That's the one I've used the most because it's less biased to clustering beforehand. But it's not a perfect solution either. And whenever you tweak your data, there's always then the risk of like biasing your data in the other direction by removing too much and so on. So you always need to keep in mind what you did to your data.
Okay, thanks.